I thank you for the kind introduction, which is maybe not correct in all aspects. I thank for the invitation to speaker, which I could hardly reject because Martin helped me to finish, hopefully, one of my last research projects, so I couldn't say no, what I do usually in such occasions. And I would like to give my tribute to the three boys, VMSE, which I have had the pleasure to work with for so long time. Alphabetic order, Werner Brannert, Franz König, and Martin Posch. A lot of the stuff I'm telling about is stemming from the cooperation with them. I will try to deal with multiplicity and estimation and say a little bit about selection bias, reporting bias, bias in admission, which is caused by regulatory requirements, something about methods of estimation to reduce bias, and something about multiple confidence interval. Now, the first scenario is very simple to be considered. I compare card treatment with a single control. I get the distribution as simple as I could have them, known variability, normal distribution with different means, my 1 to my k, and my 0 is the control group. Balanced sample size in all groups, and after a fraction of R times n observation every group, we make an interim analysis and we select the best treatment, which we go on in the second stage for experimentation, and all the other treatments are stopped or stopped for futility. And I want to quantify the mean bias and mean square error of the conventional maximum likelihood estimate of the mean treatment to control differences. Now, some word to the conventional fixed sample design when I select at the end after finishing the trial. It's correct, basically, that the kind of key final treatment versus control effect estimates in the long run are unbiased. However, it would be hiding one's head in the sand to ignore that the magnitude of the effect plays an important role in decisions and actions following such trials, particularly in drug development. The plausible strategy to go on with the most effective and sufficiently safe dose, for instance, will tend to produce positively biased estimates for the true effect size of this dose in planning the next step for drug development. Some notations, it's not very technical. The XY, say the means from the first stage after Rn observation in each group. The YJ are the corresponding means from the 1 minus R times N. The rest of the planned observation, the selected treatment. And in the end, I get an estimate Z as the selected treatment, which is a weighted average of two means before the interim analysis and after interim analysis. And ZS minus Z null is the final treatment effect estimate for the selected treatment. Now, for defining the bias, we referred to a definition long ago, which has already almost been forgotten, which has also been used by Martin and others in their 2005 paper. And it's simply, the bias is the expected value of the effect size of the selected treatment in the end, minus the true value of the mean in this treatment group, weighted by the probability that this treatment has been selected. It's a weighted average accounting for the selection. So in a sense, it gives the expected deviation from the true mean, but it does not give the weighted deviation from the targeted maximum, only from the one which is selected, not from the target maximum, which is only the same if all mean are equal, if there's no maximum. And this formula holds. We don't include the mean of the control group because this is always carried on to the second stage, and the treatment in the control group is unbiased, and we can use it, the final estimate. The mean square error can be defined quite in the same way, averaging the square of the difference to the true mean. But now we have to account for the control group because the variability of the control group has to be taken into account as well. Now, the selection bias depends on the constellation of the true means, which makes the problem hardly tractable in its general form. It depends on all the unknown parameters. Now, one of the ways to get out of this problem is that we look when is the bias largest. And this problem has been attacked in steps. There is a proof for case 2 far back in time, which has been similarly repeated in a paper 2008 by Stallard and colleagues. We have looked at that numerically for case 3, and the general proof is quite recently has been given for location shift alternatives in statistics and medicine by Carreras and Branat. Under the worst case scenario that all treatment means are equal, it's fairly easy to derive closed formulas for bias and mean square error. As you see, the bias is increasing with the square root of the time when the interim analysis is performed. R is the fraction of the information we've collected up to the interim analysis, and the mean square error has another dependence. M1k and M2k are the first and second moments of the maximum of k independent standard normal random variables. And the formulas turn out easy because we have the same mean and we don't care for the parameters. Now some results. On the x-axis there is the selection time when you look in the data. R is a half, means halfway through the sample size you look and select the treatment with the largest effect. And you see the bias increases with the time when you look into the data. And of course it increases with the number of treatments. If the number of treatments goes to infinity, the bias tends towards infinity because you take the maximum and of infinitely many random variables, independent ones. The selection bias does not increase in the same way, close to linear, but again there's the tendency it increases with the number of treatment and it increases with the selection time. So to take home, if there is a random selection at the beginning, there's no bias. One can say this is a paradigm for some of the phase 3 trials where the dose is chosen at the beginning. You may say it's maybe not random, but sometimes it may be close to the random what has been chosen. The maximum bias increases with increasing number of treatments and tends to infinity for the number of treatments going to infinity, but it's a very, very important statement. It's the one here. It sharply increases with the inspection time and is largest for the conventional fixed size sample test. And so many people say all these adaptive and selection design will have a huge impact on bias. The contrary is true. The worst design in terms of bias is the conventional fixed sample size design where selection is performed at the end, because then is the bias the worst case. It's very important to take this home. I think there's a misconception around the scientific community. When the means are differing, the treatment earlier selection increases the probability of wrong selections due to larger variability. Very clear. We have to pay a price. If we go early in the data, probability of wrong selections comes up and increases. On the other hand, the good news, if one treatment is considerably better than all the others, we have a real leading treatment, we almost always will select this treatment. We get no bias at all because we always end up with the correct maximum effect treatment and we have no bias at all. So we can go back to that situation and believe that we deal with such a thing all the time. The mean square errors in terms of the mean square error, all the figures I have shown are shown in units of the conventional mean square error if I compare one treatment and a control. A conventional 2 sample test, all the values are expressed in units of the conventional mean square error of a 2 group comparison. It does increase with k to the same, not to the same extent as the bias. It's interesting that the mean square error is identical for 2 treatments and 1 treatment, which holds under some general symmetry condition. And in terms of the conventional standard error at the end of a two-treatment comparison, the square root of a mean square error increases close to linear with a selection time R. Now there's a different form of bias, which we could look at also. Each observed effect estimate is reported separately regardless of selection, which means whenever we deselect a treatment in the interim analysis, we report the interim result, and the maximum we report the mean at the end. That's what we do. So we have a table with all the means. The selected one includes the observation from all both paths. The other ones rely only on the first sample. And again, it's defined in the way it's the expected value of the difference to the mean, to the true mean, given that the treatment is selected for the second stage, we take the total mean, and in all the other cases, we use the first stage mean. That's the definition, very similar. I don't want to show, we can also give closed formula for the worst case scenario. I only give you some take-home messages. For equal treatment means the reporting bias generally is negative, because on the one hand, if the interim effect is large, we tend to dilute the treatment effect by choosing this treatment and adding a second sample which makes a regression to the mean. It shrinks the estimate towards the mean. On the other hand, if the interim effect is small, we always tend not to select the treatment and stay with the estimate as it is at interim. So in total, we get a negative bias for those numbers reported in the figure, irrespective if they are taken at the first stage or the second stage. It's most accentuated and equal for case 2 and 3. It's strange, but it comes out like that. As k increases, the probability to be selected decreases, and therefore we more often use the hardly biased first stage estimate. So for larger k, the bias becomes close to 0. I don't want to sketch the other things. Now an interlude for another type of bias, which I call the admission bias. And I look at the situation that two identical independent trials comparing a new treatment to a control are run. I will also show the results for a single pivotal study paradigm. Each of the pre-planned one-sided z-test for the primary outcome variable at the level 0.025 one-sided has a power of 90% and an effect size of theta over sigma is 1. And now estimates are only reported or relevant for the public in case of a registration of a new drug. If both one-sided tests have been rejected, I will show you also what is changed if only a single trial is required. This will result in a bias at admission. And you can look at earlier works with regard to meta-analysis, where this formula has been considered as well by Hedges and Olkett and other. This is the bias and the mean square error. The bias is the solid line dependent on the true unknown effect size. Remember, the trials are powered at theta sigma is 1 here. And what you see is the bias is largest if there is no effect at all. These are the bad news. The good news is the probability that this happens is very low, because for the two pivotal study paradigm, this happens with a low probability of 0.025 to the square. If the treatment effect is larger, the bias decreases. Remember here, the bias is more than 2 points the standard error away from 0. Because always when you go with your estimates, you have reached significance in both trials, so your effect estimate will be more than 2 times larger than the standard error. By definition, because you ask for 2 significant results. A good news is that the bias decreases sharply, and for large effect sizes tends to 0. Clearly, there's no bias at all. The mean square error is decreasing as well, and stabilizing for large sample size, such effect sizes. It's close to the mean square error of the conventional 2 treatment comparisons. Note here, the bias for the 2 pivotal study paradigm is the lower one, is of course the mean square error is lower, the bias is the same, because we have larger sample sizes if we have 2 pivotal trials. The mean square error is lower, and the mean square error close to the planned effect size is smaller than the one of the 2 pivotals of the 2 treatment comparison study, because the distribution is truncated. There's less variability. Therefore, the mean square error tends to be a bit lower than in a conventional trial. To take home, the mean bias is larger for no effect at all. However, this is the good news, we have a very small probability to reject. It may be quite substantial for lower effect size than the planned one. That's very important. If your effect size are lower than the planned one, the bias may be substantial. If the true effect is close to the targeted effect size, the bias is small, and for increasing effect size, approaches 0 quite fast. This is the good news. The bias is equal for the single or 2 studies scenario. It doesn't help that you make 2 studies, it's only the mean square error which is reduced, not the bias. If in the single study scenario, the true effect size is slightly below the targeted one, the mean square error is slightly below the conventional mean square error because we have a truncated distribution. Now, what has been done and what has been tried to reduce the bias? One of the methods comes from sequential analysis, and it simply tries to reduce the bias by the value of the bias. And you can only do this if you have a selection rule, a clear rule in the sample space, and you are able to derive the bias as a function of the unknown parameter. If you are able to derive the bias as a function of the unknown parameter, this is the formula you calculate through the effect size. You detract from the maximum likelihood estimate, you track the bias, B delta twiddle, and you solve this intrinsic equation here, and you pretend that the solution for the unbiased estimate is the solution of this equation. This is the maximum likelihood, this is the bias, and the solution of the new estimate is the solution of this equation. It's solved by numerical iteration. And the idea is you reduce the bias by this expected value at the right side, and you achieve a reduction of the bias. This is the correction of the bias. This has been introduced, as I remember, by John Whitehead for sequential analysis, and has extended quite recently by Stallard and others for the maximum likelihood estimate when the best treatment has been selected at interim. Another way of correcting for the bias is the shrinkage estimator. I want to look at in conventional multi-arm trials. The shrinkage estimator is a weighted average between the selected maximum likelihood estimator of the selected treatment and the general mean. Now let's look how the shrinkage works. This is the overall mean. This is the crucial quantity. It is expressing the differences between the effect estimates. If there are no effect estimates difference, if all estimates are equal, the term on the right side in the red basket is 0. C dach, C hat becomes minus infinity, and you use 0 here in the left equation for the shrinkage here. You see? So what you do is, if all treatments are equal, all effect estimates, you use the mean, the total mean, as an effect estimate, as you should do, because all treatments are equal. On the other hand, if the treatments are very different, the denominator here in the red box becomes very large, C hat becomes 1, and what you do is you report the maximum like estimate for the maximum treatment just as it is. You don't shrink it. If it's a very big difference, you pretend there's a difference between the means, and you can always safely take the last one because it will be always the correct maximum treatment, and you report the maximum treatment effect. That's the idea. And it goes back to Lilly by Bayesian arguments and Wang, who have given modification quite recently in statistics in medicine. Carreras and Brannert extended this for 2-stage design with selection. It's a fairly straightforward process. They used the second independent sample as an independent sample. They apply all for the first stage and use the second stage sample as an unbiased sample and combine those estimates and try to find improved estimates which reduce the bias. Another way of correcting for the bias is the uniform minimum variance conditional unbiased estimate. And this type of estimate only uses the discrepancy between the largest and the second largest mean for shrinkage. It's not unusual, it's not unreasonable, because it matters how different the largest and the second largest is. If there's a large difference, you can safely assume that the largest one is much larger than the other ones, and you can take it as it is. So it's not quite unreasonable, and I think Bauer and Klim quite recently have extended this to the heteroscedastic case and the case to select the second largest and so forth. Bias correction to take home. How to define bias in case of selection? This is the first question. It may not be uniformly agreed on how we should define bias in this situation. What really bias matters? Bias or mean square error? An old discussion. Is it the bias we need? Is it the mean square error? What matters? What is a suitable criterion for a good estimate in the end then? I think a very important question. And I don't speak about exception, I speak about the normal trials where more than one treatment is involved. This is daily life. We don't speak on extra models, complicated design. We speak on daily, normal things we observed every day. The next question to the regulator or to us is, would we always also reward conventional estimates? Who would regulators ask for it anyway? My private answer is, of course, everybody reports the maximum likelihood estimate. I think nobody would give a bias-corrected estimator without at the same time giving the maximum likelihood estimator, but I think this is a matter of discussion. Generally, correction and shrinkage becomes larger the more similar the effect estimates are. Clearly, because then we believe that we have to correct because there should be a bias involved by selection. Now, the next question to the regulators, to the companies is, should it be tried to give bias-adjusted estimates in the spirit of a sensitivity analysis? We often ask for sensitivity analysis. Why should we not give bias-corrected estimates for some sort of sensitivity analysis? Now, a few words on simultaneous confidence as well and stepwise multiple test. It has been addressed several times this morning that, contrary to the conventional non-stepwise test, for stepwise multiple test as the home procedure, compatible simultaneous confidence intervals, which means rejection of the multiple test procedure is non-coverage of the hypothesis by the multiple test procedure, are not straightforward. The form of the confidence interval depends on the choice of the set hypothesis. We have seen an example of skewed non-symmetric confidence in the morning. There may be a situation where such a special choice is advantage. I remember the situation when we looked at testing non-inferiority. So we defined a family of one-sided null hypothesis above 0 and low 0. And we could use one-sided level alpha tests, 90% confidence intervals, for the price that the point of equivalence is always contained in the confidence interval. So the only price we pay is that we leave out the 0 point from multiple testing. We always have a confidence interval that covers the 0 point, and we can instead use a 90% confidence interval instead of 95%. This may be an advantage because the family has a special structure. Compatible simultaneous one-sided stepwise confidences are ending at the null hypothesis. For instance, you get intervals like from 0 to infinity for the least significant treatment effect, thus only reflecting the stepwise decision. The concept that such new simultaneous confidences add only information when the corresponding null hypothesis is not rejected in the multiple test is questionable, particularly when the rejected null hypotheses are the ones which are triggering the further action, as usually in the regulatory context. You are interested, where are the estimates? If I come up with a significant effect, you're clearly interested, where are my estimates, not of those who are not significant. It's for the cost effectiveness, for assessing the benefit of the treatment. It's important to get good estimates where the effects really are, and not of those which you never would register anyway. And maybe the focus on the yes or no decision in the regulatory context have produced these confidence intervals, which reflect simply the yes or no decisions. If you ask me, I would say it adds another issue of multiplicity if we sacrifice the equivalence of confidence interval and multiple testing. I would not use confidence intervals which are different from the multiple test procedure, because this adds another thing of uncertainty, of another thing of complexity. I think, to my opinion, I wouldn't keep up that equivalence. And my last question is a little bit provocative. It says, does this question a lot of sophisticated work on simultaneous confidence intervals or even on multiple testing to be applied in other areas other than a regulatory context? Note, however, to be sure, that the phenomenon of bias has to be considered as an intrinsic factor of human life when selecting jobs, friends, partners, based on a comparison of past observation, which are afflicted by random variation. Thank you very much.
Thank you very much, Peter, and particularly for the non-statisticians in the in the room. The last slide tells us all that we understand this problem more than we actually realise. Thank you. If you can step down, Peter, then I'll ask Brian Austin Millen to step up, please. Brian's going to lead the discussion on Peter's presentation. Bias from Eli Lilly. Brian, all yours, please.
Thank you, Rob. Thank you, Peter, for that talk, and I want to thank the organizing committee and Amelie in particular for the invitation. At the close of Peter's talk, he touched on a very important topic, and that's the one of the availability and utility of confidence sets that correspond to stepwise multiple testing procedures, which we commonly use in our clinical trials. It is on this topic that I'd like to offer a comment for the next few minutes to kick off the panel discussion. The problem of defining confidence sets corresponding to multiple testing procedures is one that's been studied for quite some time, whereas decades ago it was felt that confidence sets were not available for such stepwise procedures. The predominant wisdom and the result of work of 20 plus years lets us know, one, that mathematically appropriate confidence sets, simultaneous confidence sets, do exist, and two, that these are generally non-informative. By non-informative, I mean, as alluded to in Peter's talk, they often simply reflect the information that's already provided in the binary reject or fail to reject test procedure, at least for the rejected hypotheses. In fairness, though, I must point out that that the statement that the confidence sets are non-informative is only almost true. Just very recently, Jason Shu, who many of you may know, pointed out to me that in a 1994 paper that he and a colleague offered confidence sets for stepwise procedures that could offer additional information beyond just the reject hypothesis conclusion in some cases. The rub, however, is that the existence of the informative confidence set is results dependent. For example, if all hypotheses are rejected, then we may get more informative confidence bounds. Olivier Jabot also shows this in his recent paper where he addressed confidence sets for the Holm, Hochberg, and Hamel procedures. So the issue of having confidence sets is still a question. And so it leads us to, well, is there another way? And to this, I think there are two answers that we should discuss here today, and I'll kick over to the panel. The first one is conceptually straightforward per recent research, which includes some individuals in this room. And the second one is probably a bit more provocative, but it's a position which I think reflects the practicalities of the regulatory and approval process. Well, the first answer essentially says, well, you can get more informative confidence sets corresponding to these common stepwise procedures, but at a cost. And the cost is reduced power. So if you're willing to accept less power to reject as many null hypotheses as possible, you can get these more informative confidence sets. Several authors have offered such an opinion. Speaking as both a trial sponsor and personally as a potential clinical trial participant, this is not a trade-off that I feel is acceptable in principle. It's my belief that we owe it to clinical trial subjects to make the most efficient use of their sacrifice to participate in clinical trials. From a societal perspective, I don't believe that reducing the power of a trial or reducing the probability that a trial will conclude benefit when it actually exists is I believe that is unacceptable. Similarly said, requiring more subjects to be exposed to experimental conditions than otherwise required. To have the same power is both costly financially and ethically problematic. So while the methodology exists to get these more informative simultaneous confidence sets, I think the obligation of the trial sponsor, and thus the statisticians, is to use the most powerful testing procedures that are reasonably available. Well then, if one is to reject the advances and their practical application to clinical trials, where does that leave us? Is there another way to provide this useful information? And here, I think it's helpful to think not just through the lens of statistical science or through the lens of a statistician, but also through the lens of a clinician or other regulatory scientists with whom we collaborate. If we statisticians are generally happy and we're generally convinced that our conclusions based on our multiple testing procedures are solid, why is it then that our non-statistical colleagues yearn for and request confidence sets? My perception, my experience has been that they do this for a couple of reasons, and the primary is to understand and have some sense of magnitude of effect. And then armed with this estimated understanding of the estimated magnitude of effect, he or she is then likely to do some internal meaning to that person, internal comparisons, perhaps to a threshold of what he or she would deem to be clinically relevant, perhaps to a threshold that he or she understands to be the effects of other treatments that are currently available on the market. And then, very, very importantly, overall, regulators are going to take the estimation information and use it to get to an overall sort of subjective benefit risk conclusion or benefit risk assessment. It's important here to note, as we think of benefit risk, that this generally involves all the data. It's not limited to just the data that were included or just the endpoints that were included in our multiple testing strategy. Endpoints across the gamut are included, and indeed non-significant results are included as one attempts to form some opinion about benefit risk. So overall, that leads me to the conclusion that the uses and the reasons that are for our colleagues wanting confidence sets are somewhat divorced in their minds as they try to reach conclusions of benefit risk from the question of statistical significance, which is, of course, the primary threshold for determining efficacy. So that said, as we move to the panel part of the discussion, I think the following question is worthy of consideration, which is, do unadjusted confidence intervals have any role to play, even in the face of trials with complex multiple testing schemes? I think Peter had started to allude to this. I'll offer just a couple of comments, and then I will turn it over to the chair. My view is that unadjusted confidence sets do offer some additional information, albeit imperfect. We naturally now, and the tension that is created is naturally exists now as we side by side and juxtapose multiplicity adjusted p-values and naive or unadjusted p-values, both in publications and in regulatory submissions. And I think the use of the unadjusted confidence set actually reflect the reality that regulatory approval is not equal to statistical significance. It is there is a bit of an art to the science. And lastly, I propose consideration of this somewhat controversial idea because I have faith that our statistics community, that statisticians are very well equipped to collaborate with non-statisticians and help us overcome the interpretational challenges that would exist, of course, with having confidence sets that do not directly align with the multiple testing procedure. With that, I'll turn it back to the chair.
Thank you very much for that thought-provoking discussion. So I'm going to ask in a moment Kathleen, Amelie and Norbert. I guess you don't need to introduce yourselves. You've all had the microphone already today. But I'd ask you to give a reaction to the presentation and the discussion, particularly on these key questions which have been outlined by both Peter and by Brian, and ask a question back to the speakers if you wish, and then we'll open it to to the floor. Whilst you think this is an aside for the non-statisticians who heard this morning that not all p-values less than the nominal 5% level can be trusted, as you'll get some false positive claims, and have now heard that estimates and confidence intervals are also unreliable. It's becoming murky, isn't it? So, who'd like to start? Kathleen, your name is first on my list. So a reaction and, if you like, a question, please, just shortly, and then we'll open it to the floor.
So I think there is a lot of challenges. I think I'll talk mostly about the issues of confidence intervals. I think people are hungry for confidence intervals in these settings, but I think it becomes really challenging. Like we've noticed, I think a lot of people are really not aware that there are confidence methods for these, and if they were aware and they saw confidence intervals that look like 0 to infinity, they probably wouldn't believe the statisticians that that actually is considered a confidence interval. But even I find a lot of statisticians are completely unaware that there is a way to do this. And it also becomes challenging, I think, then, like when we talk about labeling. So as I mentioned earlier, we may have one set of endpoints that we put under our multiplicity adjustment, but that may or may not match what goes in labeling. Well, how do you translate the whole paradigm? I'm sure, does it start to fall apart when we start picking and choosing endpoints to put in labeling? How can we interpret that? I think most people know that unadjusted confidence intervals, they're a little uncomfortable with that. That's too good. Bonferroni are too wide. That kind of loses everything. I think it's a really challenging area. I'd love to see if we can make advancement on that, but it causes a lot of challenges.
Thanks, Emily.
Yeah, I think if we have the possibility to give a simultaneous confidence interval, we should as well report it. And my question for Peter is regarding the assumption of equal treatment means. I think in clinical studies, usually this assumption might not be fulfilled because we will have other kind of treatments, means with different doses, for example. And the question is whether Peter or his group has any numerical results for the selection and reporting bias in realistic settings where this assumption is not fulfilled.
Well, when we started the work, we started to simulate all the bias. And it's possible, of course, to simulate the bias. And when the referees wrote back, well, it's quite interesting what you present there, but it's rather of low value because it's all simulation. We sat down and tried to find out the explicit formula. If you have no equal means, the explicit formula are hardly accessible because the maximum, distribution of the maximum becomes very easy if equal means. And if you have unequal means, it's a messy thing. But you can always use simulation. But the problem becomes very complex because you have k unknown parameter, and it will be very difficult to interpret. But it's in principle, of course, it's possible. Let me say a little bit on the confidence decision. I mean, the scientific community has moved away from p-values. When you today write a paper in a good medical journal, people tell you, don't report p-value, report estimates and confidence intervals. That's the tendency it's existing. And this is certainly is the reason that the yes or no decision is not that important in a general scientific paper. The regulatory context is specific because the game is to make it. The game is, it's part of the game to make it. And of course, statements like it's very important to get the best way, the maximum power to make the game is clear in this context. But that doesn't solve the problem that you would like to have information on the type of estimates. I mean, in the effect, cost effectiveness and all the things which will be more important in the future have to be assessed by looking at the estimate and not on the fact that you rejected the hypothesis. So I don't say it's an easy issue, but I would say testing is certainly not all in this context because there are many other things combined with registration of a drug.
And Norbert, please.
Yeah, well, two comments or questions to Peter. Well, you talked about the conventional trial where the bias is highest, the selection bias, the conventional meaning that you don't have an interim. But the very conventional trial or the very conventional procedure is having a dose identified before you start the phase 3 trial. So there would be no bias. So is this, could this be a reason to going back to the very conventional setting where you have clearly separated the exploratory phase and phase 3 is just to confirm the finally identified dose and regimen? I guess it's probably a bit too harsh, but this was the procedure we followed in in the very past. So then it probably has to do with the extent of bias we would expect, and from my feeling, this should be assessed what the extent is. It's probably not as important, but okay. Question. The second question is if. We have a selection bias in the one direction, reporting bias in the other direction, and admission bias again in the direction of the selection bias. Then we report our estimates at the end of the day, and we take the benefit risk from these estimates. Then we may overestimate the positive effects of the benefit. And the question would then be: is it always okay to rely on our, let's say, common sense clinical judgment on the estimates we see, on the data at hand? Would this be a reason to really incorporate secondary endpoints that are relevant for the risk benefit in the confirmatory strategy? Because we can't rely on the estimates because we may have a bias that overestimates the benefit risk.
I would like to answer the last question in my second life. The first question, I think we have a sentence in our paper where we say the only trial which creates no problem with multiplicity and bias is a non-sequential single treatment control comparison. I think we question the same way as you say, is this a sign for the way back, to have no problems at all, to avoid any interim analysis and give, but I think this is not the answer. We know that this is the trial which have no problem with bias, with no problem with estimation, no problem with multiplicity of treatments and so forth. But I think this is not, it's not the answer that we should go back to that because it has other disadvantages. And I think the question you asked, I can't answer all these other questions. They are too complicated for me.
So comments or questions from the floor, including answers to Norbert's complicated questions, if anyone would like. Or Peter's rather straightforward questions, at least rather straightforward to ask, rather complicated to answer. What's the criterion for a good estimate? Comments or questions? Yes, please, Willie. Maybe you introduce yourself briefly as well.
I'm Willy Maurer from Novartis. I have a question regarding unbiased estimates and confidence intervals. You presented some methods to compute bias-reduced or even unbiased estimates. Are there some methods also to sort of compute confidence interval that relate to these unbiased estimates?
I think there are some, but I don't want to pretend that I can even give you any details.
Any others on that same question? We have a number of leading thinkers on this topic in the room. Other questions then? So I want to ask a difficult question to the panel. And again, I'm going to focus on the sort of non-statistical angle of this to some extent. So there seems to be some agreement on the panel that, let's call them conventional estimates and confidence intervals, have the potential to be unreliable and potentially even misleading for decision making. And so what people will want to know is the extent of that problem. To what extent may estimates and confidence intervals be unreliable? How much can these different types of bias impact the reliability of the decisions that we're making based on these data? How do we quantify what the problem is? How big a problem is it in reality? Perhaps Peter or Brian?
So Rob, the question of essentially, I think your question was one of quantifying bias. Is that right?
Yeah, but it's very difficult, of course, because I'm trying to get people not to do it in statistical terms, but to move on and tell people who are actually making the decisions what's the impact on the decisions they're making of this problem that's being described.
Yeah. So just a couple of comments. I think it's hard to convey the exact impact on a decision process, largely because the decision-making framework is not really one that's transparent. We certainly know how to evaluate statistical significance, and we know that as a contributor to the framework. But we also know that regulators, again, sort of apply a bit of art to making some global benefit risk assessment. And without truly having that framework be visible, it's certainly difficult for anyone to then sort of quantify the impacts of bias, et cetera, on that framework.
Any others?
Well, Peter. I think the good news is also in the admission bias, which for me is a little bit the most striking thing, when this is a very simple calculation, and it shows particularly the danger if you have an underpowered study, but you still succeed and your effect size is in fact smaller than the targeted one. Then in order to get a significance, you must have a high mean, and then you tend to overestimate your effect. And in all these situations where you have not the effect size which you had in the planning phase, and these are bad news, definitely. If you are always pessimistic and you are beyond your assumptions, you have a very good effect, I would have no problem. And the calculation shows if you have really be careful in planning and your effect is better than you have planned, then there's no problem. The problem is if your targeted effect size is by no means reached, and we know often this is because people are optimistic, samples are expensive, and with a certain probability you get the game, but then you must expect that your bias is quite substantial, because you would never reach 2 times significance or 1 time significance. That's by law. That's by definition of our requirements for registration. Nobody can do against. This is the rule. This is the game, and we have to face this. Just as it is the game, as it was in older times, people made 5, 6 trials, and they went to the regulatory agencies when they solved. It's a good time now. It's sufficient. That's a sequential design that was overly biased in any means, and I think there are many, many examples where the effect estimates at the registration were overestimated and were shrunk down later on by practice.
Any final questions or comments? I mean, I conclude from all that the panel have said that there are clearly some issues to think about here, and it's also clear to me, at least, trying to answer my own question, that if I just look at the first one of those, selection bias, that will have a fundamental impact on the estimated effect of treatment in a particular circumstance where you're making a selection decision. So these are non-ignorable problems. There'll be some other situations where we are talking about rather small and I would say inconsequential differences on confidence interval widths, but there are some rather fundamental situations that are being described here. some methods proposed for bias-corrected estimates and some thought about confidence intervals to attach to the relevant tests, and a proposal that they are useful to be included in some way when analysing and presenting clinical trial data. And the final reflection is to pick up on Brian's insightful comment that potentially some of our solutions to these problems come at a cost, whether that's a statistical cost in terms of power, or whether that translates into a cost in terms of resource, most importantly, the patients in the trial, but also the delay in time to market for beneficial medicines. And to me, and you'll have guessed it from my comments earlier and my questions earlier, but we have to balance both sides of this equation, both the sort of statistical rigor on one side, but also the cost of getting the benefit of that rigor, which may sometimes outweigh